Our last genetic problem video will be on X-linked traits, colorblindness, hemophilia, multi-allelic traits. We're going to look at blood type and trihybrids. So let's go ahead and get started. So if you look at this problem, and we are talking about X-linked. If you look at this website, it's pretty good. So we're going to be looking at X-linked. A boy is colorblind. And they say use B. Here. So he is colorblind. His parents and grandparents had normal vision. We know he got the Y from the dad. So we know we had to get this one from the mom. So the mom, draw these in different colors. We know the mom gave him the little b. So we know she has a little b. But she has normal vision. So let's make that. So we know she has normal vision. Oop, didn't change over. So we know that this is her genotype. His father, another color for him. Let's see. His father gave him the Y, and he has normal vision. Oh, he has normal vision. So we know that he has a big B as well. So I did the parents, now I have to do the grandparents. Grandparents have normal vision. So let's look at her father. So I'm going to write right over this. So her father had normal vision. That's her father. We know he had a big B. Let's check out, see what her mother is. So her mother XX had normal vision. So we know she had a big B. But we know that she got this little b from here. Phenotypes for his mother. One, mother. His maternal grandparents, right over here. There they are. And that's his father. Humans, brown eye is dominant to blue eyes. So look at that. Take a picture of it or something. And this will also be on your lab exams, things like this. Humans. Brown eyes. Let's see a brown color. It's kind of brown. So brown. So big B is going to be brown. Alright, I'll just put it over here. And this is going to be recessive. Alright? And a man who is colorblind. Like up there. Yep. So he is colorblind, just like up there. Alphabetical order. And I don't really like using this B, all these B's. Maybe I'll change this. B. I don't know what we should do. Let's make these C's. So we're going to use C's up here. I'm not going to redo everything. So brown. So we'll keep our big B's for them. Little B's here. Colorblind. And he is heterozygous for brown. Alphabetical order. Heterozygous. Right? Okay, so the blue-eyed woman. So we know she's this. And she's a carrier for color blindness. Let's use the blue for that trait. Nope. And she is a carrier. So that means, there's my green. I'll just use green for this trait. She's heterozygous. Alright? So this is his genotype, that's her genotype. Two sons who are colorblind. And one has brown eyes and one has blue eyes. Alright, so they have two sons. Blue is the color for the gender here. So two sons. I'll just put S for sons. And they're colorblind. And it's important for you for your test so you know how to figure these out. Oh, I was going to make those C's, wasn't I? Alright. And she is a carrier. So she has blue eyes, colorblind. See, I did it again. And she's a carrier. Alright, they have 2 sons. And it says they are both colorblind. And one has brown eyes and one has blue eyes. So let's say this one on here has blue eyes. And then it says one has brown eyes. How do we know what the second one is? Well, look over here. We know what the second allele is there because the mom has blue eyes. So we know it's heterozygous. And they have a brown-eyed daughter with normal vision. Something has happened here. So they have a brown-eyed daughter again. We know, so here's the daughters. Brown-eyed. And we know that's heterozygous because the mom has 2 little bees. Can only give little bees. And we know it's a daughter, so it's 2 X's. And normal vision. So let's take a look at what we can figure out here. The mom's a carrier. She has normal vision. So we're gonna have to assume that she got that big C here from the mom because let's go here. She got that little C from the dad. So right here, she got that from the dad. She did not get a Y, she got that. That's right here. Alright, we're now going to go to another page. And you have this page in your handout. I already have this one done for you, so let me go to my GIMP board. But what we're going to be looking for is albino and normal pigmented. So pigmented, that is going to be autosomal recessive. And hemophiliac is going to be X-linked. So it said we had an albino and normal. So we're looking at pigmentation. And I did this A is normal. Oops, there should be an L there. Color. So this is going to complete dominance trait. And then we have hemophilia. This is X-linked, so we're going to be writing it on the X's. And it's going to be different between the males and the females. So go ahead and look and see what I did. So females, these are X's. There's two ways to be normal. Homologous dominant, heterozygous. Only difference for the females is that you write them on the X's. And this is a carrier. Oops. Not very good word. Go ahead and look and see what I did. So make sure you know why males cannot be carriers. C-A-R-R-I. It might be a little too thick. Carrier. Females are carriers. They don't have the trait, but they can pass it on. So the heterozygotes are the carriers. Notice in X-linked, males do not have two of the alleles. This is X-linked. They only have one X. Since they only have one X and the other allele is a Y, they cannot be carriers because heterozygotes are carriers. So nothing goes on the Y. So if you look at this, XH, that comes from the mom because the Y comes from the dad. Hemophilia, females need two little H's. Males only need one. So that is why there are a lot more males who have hemophilia than females. Females need two, one from each parent. So let's look at the sons again. Cannot be carriers. You have to be a heterozygote. There are no heterozygotes in the X-linked traits. They only have one allele dealing with this trait. The other allele are Ys, and they come from their father. The X's come from their mother. So if they're colorblind or have hemophilia, they got that allele from their mother. And I am going to be taking a shortcut in here. Shortcut. Oops. Because I have homozygous. And I already did it all for you. So if you want to stop and fill yours out. Again, I did take a shortcut, which makes it really, really, really nice to figure out the different kinds of sons and daughters that are possible. So go ahead and finish. Fill it out. Alright, so after you fill it out, what you have to do is figure out what are the different phenotypes for the daughters and what are the different phenotypes for the sons. Which is really nice when you can take a shortcut because now you only have one column right up here for possible daughters and the other for the possible sons. And in this example, the types of phenotypes are going to be the same between the sons and the daughters, but that is not always the case. I'll make that a little smaller if you want to take a picture of it. Oops, went away. So basically, it's pretty much the same, except for those two traits, only put hemophilia and colorblindness on the X. The main difference is with the males because they only have one X. Alright, let's go ahead and do our next one then. So same page. A colorblind man So just like the hemophilia is going to be on the X, a blood group O marries a normal vision woman of blood group AB, whose father was colorblind. If they have 4 sons, what will these sons most probably be in regard to vision and blood group? What if they have 4 daughters? So again, I did this all out for you already. So let's go to my GIMP board. Let's review blood type right up here. We have A blood, AB blood, B blood, and O blood. A blood, AA, and AO because O is recessive. AB blood, codominant. B as BB or BO, and O blood, OO. For blindness, just like hemophilia, let's make that a little bit bigger so you can see it better. Here's the females. Here's the carrier right there. Remember, males cannot be carriers. They get the X's, so whatever the mom has on the X, that's what the son's going to get. The Y's come from the father, the X's and the sons come from the mother. So if the son is colorblind, he got it from his mother. And then it says a colorblind man of blood group O. Here it is right here. And he's a man right here, XY, and colorblind, little c. And then we have a woman of AB blood, alphabetical order, even though it may not be written in order. And normal vision. Whose father was colorblind. Right here. Just like this. If her father's colorblind, this had to be his genotype. So she had to get a little C from the father because she did not get the Y. Otherwise, she would be he, and she's not. So now I run across those, and again, I am doing a shortcut because I have 2 O's. Go ahead and try to figure it out. Try to do it on your own, and then I'll show you. So if you're watching this, turn it off. Go ahead and figure it out. So here it is. And it's really nice having the shortcut because I have all the possible daughters in one column and all the possible males in one column. So these are the type of daughters I can have. These are the type of sons I could have. And this is again a case where they're going to be exactly the same. A normal, A colorblind, B normal, B colorblind. And it's going to be the same for both genders. Now we're just going to do the trihybrid and then we'll be done. Let me make it smaller so in case you want to take a picture of it. The whole thing there. Let's see. There you go, in case you want to take a picture. Onto the trihybrid. So on to the trihybrid. Whenever you do a trihybrid, we're always going to be using this method. Always. So suppose that R1 is red. Over here. I did it so it's alphabetical order. And let me make this a little bigger. And let me use a different color. So R1 is red. R1, R2 is going to be pink. And our 2 is going to be red. And it says that statement here, right there's a statement. So look, we have, what do you think this is? Complete dominance, incomplete dominance, what is it? So both of these are incomplete dominance. How do we know? We have 3 different phenotypes, and they're writing with R1s and R2s. So let's say this is red. What do you think the heterozygote's going to be? Oops. Purple, right? That's how you make purple, red and blue. Now what I'm going to do is I am going to do a monohybrid cross with these over here. Cross a blue and pink plant homozygous for green. Let's go ahead and do that one. I'm going to get a darker green here. Try this color. So let's see what they're going to look like. Let's do this one. A blue. We know it's B1, B1, right? Pink. But we gotta do it alphabetical order. And homozygous for green. And then pink. What's happening here? So pink. That's our first one. And it's going to be crossed with one that is well, I'm going to go ahead and stop this and see if you can figure it out, alright? So we got. Now what I'm going to do is I'm going to be doing 3 monohybrid crosses. And I'm going to do my first cross with the bees. Alright, so these ones, and look, I can do a shortcut. I am going to be taking that shortcut. The bees right here. So do 1 monohybrid cross with those. Then go and do another one with the G's. And then do a monohybrid cross with those and get their phenotypic ratios, and then we'll go back together after you do that. Go ahead and do that. And so here it is. I did 3 monohybrid crosses. Notice I took a shortcut here and there. So here I took a shortcut. And here I took a shortcut. So 1 plus 1 is 2, so 50-50 chance blue or purple. There was only 1 recessive allele for the green, so there were no yellow, so that line died out. And the third right here, incomplete dominance, red, white, and pink. Red, pink, and white, I should say. It was in a 1 to 2 to 1 ratio. 1 plus 2 plus 1 is 4. That's our denominator. You can use decimals if you like. And then you got to make sure you add all the numerators together, and you get 16. So this was a trihybrid. And let me try to get it on one page. You can take a picture of it if you like. So there it is. And that's all the genetic problems I'm going to be doing. Hope it helps.